Thank you very much to the JGI to invite me here, especially since we are still a reasonably new group at UC Davis. We started out about 2 years ago, so I'm really excited to share some of the work that we've been doing since with you. And Axel already mentioned it. What my lab is focusing on is the discovery and to some degree also the engineering of plant terpene metabolism with the idea of using this knowledge for new tools for bioproduct manufacture, as well as, for example, improving crop resistance. So I will span those both worlds a little bit today. So what I would like to do today is to tell you a little bit about the ecological and economic relevance of terpenes and specifically diterpenes, so the subgroup of higher molecular weight terpenes that I will be focusing on today. I speak a little bit about some of these tools that we are applying to investigate terpene metabolism in both non-model and model species, and then just give you 2 little vignettes on applications that we have now found where we could use these tools both for the discovery of new diterpene pathways for diterpenes as potential drug lead compounds, as well as diterpenes that are involved in particularly pathogen resistance in bioenergy and food crops. So why diterpenes? So really our interest in diterpenes stems from their versatile and very potent biological activity. Most of you will know gibberellin phytohormones and the phytol side chain in chlorophyll. These are actually derived from the diterpene pathway and have obviously critical functions in plant development. But the much larger fraction of the diterpenes is involved in these type of biotic and abiotic stress responses, often very species-specific. We have several well-established examples, for example, in the coniferous tree. So the sap of the coniferous tree is almost exclusively derived from terpenoids and diterpenoids that are used against pathogen and insect defense. But also in many of our food and energy crops like maize and rice, they have very prominent diterpene groups that play a key role in defending the plants against pathogens in particular. And sort of a picture that is emerging more recently is their function in signaling. There are a few papers out now. Sort of traditionally, diterpenes and terpenes have been seen as toxic compounds primarily involved in defense, but we're seeing more and more examples that sort of hint at that there's more to this story than toxicity, and that there actually might be more of a function in signaling in plant-environment interactions that these compounds contribute to. And so this biological activity is also really the foundation for their industrial importance. So the second sort of reason why we're interested in it is the industrial uses. Diterpenes in particular are interested in the pharmaceutical industry. The most prominent example certainly is Taxol, the most widely used anti-cancer drug that alone has about a $2.5 billion annual market. But also new drugs that come on the market like the CM booster forskolin, or new drugs here like Salvia, a compound that I will speak about just a little bit later. But in addition to pharmaceuticals, diterpenes are used in the fragrance industry as a base note to a lot of the high-value perfumes. Natural sweeteners like steviol are terpene-derived, many other functions. So taking these 2 factors into account, their physiological as well as their industrial relevance, this is really what motivates us to look into how can we actually elucidate and get a better picture of this diversity of diterpenes that spreads about 12,000 compounds across the plant kingdom in enormous structural and biological diversity, many of which we find in non-model systems, so systems where we do not have any genome resources, any transformation tools available. Only a few of them, like here in rice and in maize, where we have model systems that we can actually use to study some of these biological functions. But for the most part, we are dealing with non-model systems. So when we started the lab, the first thing we had to do was to find better ways on how we can efficiently discover diterpene pathways in these systems. And what we sort of tap into is the fact that despite their diversity, all of these diterpenes, more than 10,000 compounds, derive from a single central precursor here, general diphosphate or GGPP, which in turn is a product of the condensation of these 2 central 5-carbon intermediates, DMAPP and IPP, that are the precursors to all of the isoprenes. And now from this single precursor here, there are essentially two enzyme families, diterpene synthase that catalyzes cyclization and isomerization of this linear compound here, GDPP, that can generate a number of different scaffolds, mostly polycyclic scaffolds, but also linear ones. And these then are taken on by the superfamily of the cytochrome P450s, mostly located at the R, that now will attach a variety of different functional groups, starting usually with sort of single oxygenations and further carboxylations, other functional modifications, to eventually arrive at this large number of more than 10,000 compounds. So we're really honing in to these two major enzyme families that together really drive the chemical diversity of diterpenes and terpenes in general in plants. And it sort of relates back to what Betsy just told you in the previous talk, is both of these enzyme classes are part of these superfamilies, especially the terpene synthases, where we have a large diversity that is built on a high structural similarity and a high sequence similarity across the family, yet a very large functional diversity. And just sort of highlight this for the terpene synthases, really as little as a single residue exchange in the active site can change function in the terpene synthase. So computational prediction is rather difficult, which for us means we always have to go the way of biochemical characterization. And wanting to do this efficiently, we had to sort of find a way on how we can actually look at a variety of different terpene synthases in P450s and go after their functions. And so this sort of brings me to some of the tools we've been developing in the lab, looking at particularly non-molar systems, but now we're actually starting to sort of go back a bit and look at some of the reference species. But what we usually start out with is metabolite profiling, GC or LC-based metabolite profiling across the tissues of a plant or across different inducibilities or developmental stages to identify those conditions and tissues where we find these diterpenes to be most abundant. Obviously, we have to take into account issues of transport. Oftentimes, we sort of ignore this in this first stage. But this is a very important part for us to be able to sort of hone in onto certain tissues that we can use with a good idea that we have an abundant pathway there. And what we do then is sort of standard transcriptome sequencing, getting the assemblies. And what we have then developed is essentially sort of our own in-house pipeline to efficiently find gene candidates for terpene synthases and cytochrome P450s. And sort of we have a combined database where we first look at domain features, classic domain features of these enzyme classes, where we can very quickly filter a large assembly to a certain subfamily. We then look into gene to metabolite correlations, again, looking at different developmental, inducible, or tissue ratios. And lastly, we'll do phylogenies, looking at what still is somewhat a limited number of known functions to, in the end, efficiently get to our enzyme candidates then for biochemical characterization. And so this really allows us sort of from the stage of having a final transcriptome assembly to our enzyme candidates within the day. And that was really helpful for a new and a fairly small group to go through a number of species efficiently. And so this is what we have done so far. We looked at some 15 different species, anything from bioenergy relevant crops to medicinal plants, as well as plants with some relevance for fragrance production. What we found across these, using these tools, is about 70 of these diterpene synthases and some 600 cytochrome P450s, already narrowed down to those P450 subgroups that we know can have functions in terpene metabolism. So this is a start, but this is still a large number of enzymes to go through with biochemical characterization. So we have sort of evolved what we're doing over the last 2 years. We started out with sort of the classic way of heterologous expression in E. coli of these enzymes, affinity purification, and in vitro assays, which works really well, but it's fairly cost-intensive for the commercial substrates and the assay conditions, and it's also quite slow. So mostly what we do today is actually co-expression analysis, both in Nicotiana benthamiana as well as in yeast and E. coli, with the collection of terpene synthases and P450s we already have, and we can throw new candidates into these systems and really efficiently use these multi-gene co-expressions, extraction of the products directly from the media or from the plant tissue, and then go after new metabolites, new products via mass spectrometry and NMR, usually in combination of the two. And so we've used this for a number of species now. We have probably about 60 diterpene synthetases characterized so far, and a few P450s. They tend to be more difficult because they're membrane bound. But it's been quite efficient to use this tool to find interesting new chemistry across this terpene synthase and P450 superfamilies. And so using these tools, what I just want to show you today are two small examples on how we have used this. One example in a non-model system where we are looking into this compound here, salvinorin A, in a medicinal plant. And then another example where we find new diterpene pathways involved in maize pathogen defense. So starting out with this non-model system, the plant in question here is Salvia divinorum, or diviner sage. It's actually, most of you will maybe know it as a recreational drug. It's been in the news quite a bit. But what really interesting is that the major compound, the hallucinogenic compound of this plant here, salvinorin A, has actually quite interesting medicinal purposes. And what it does is it actually binds to these opioid receptors much alike morphine or other alkaloids do. Right? So it has very similar bioactivities in its binding properties, but what it doesn't have is any addictive properties. So thinking about this is a very interesting target, for example, for the treatment of drug addictions and other neurodegenerative diseases. And so salvinorin A has just actually entered clinical trials for these purposes. And so we were very interested to find out how this compound here is made and to start elucidating the biosynthetic pathway. So the first thing we did was looking across these tissues. Salvia divinorum is a member of the Lamiaceae, so the mint family. Typically, we find these compounds highly enriched in the leaf trichomes in these species, and indeed, this is what we found here. So here, this is simply a cotton swab of the underside of the leaf, and you can see salvinorin A is highly abundant in these trichomes. So we isolated a number of trichomes. One of the undergrads in my lab spent some weeks of isolating enough trichomes to isolate RNA from them, and we could sequence those, and what we found was two enzymes here, two enzyme candidates that we believed could be potential candidates for this first step in the production of salvinorin A. We characterized both of them. One turned out to be an end-copolyl diphosphate synthase, so a precursor for ghrelin phytohormones that left the second one. And this indeed showed us a new function. So this here, F, is the major product that we found. These other products are sort of typical byproducts that we usually see when we analyze these type of diterpene synthase products under JGI because they degrade very quickly under the heated conditions of the analysis. What we could combine it with LCMS and NMR, and we could verify that this enzyme indeed converts our central precursor GGPP through a number of carbocations into this novel intermediate here, colavinyl diphosphate, which has a very characteristic methyl group arrangement here at the decalin ring. And if you compare this to salvinorin A, this indeed now is sort of the first committed precursor on the biosynthetic pathway to salvinorin A. So it is only the first step. So we still have a lot of open questions to address, which primarily will be cytochrome P450 functions that will now decorate this decalin ring here with these different hydroxylations, acetylations, and so forth. And we're also interested, since diterpene synthases are such a large superfamily and structurally so related yet diverse, how these potentially have evolved from their precursors and progenitors in ghrelin biosynthesis. And so what we did was, using homology modeling, which is really very straightforward, because these enzymes are structurally so related, combined it with ligand docking here of the GGPP in the active site, and then looked at functional residues in the active site in about 6 to 8 angstrom radius around the docked ligand, and took then those residues and compared them to all the diterpene synthases we had characterized so far, and could sort of pinpoint some unique amino acids for site-directed mutagenesis. And so a graduate student in my lab did this work, and what he could do is actually he could reprogram this enzyme here, the CPS2, from its wild-type function into 4 different functionalities with as little as a single residue switch. one thing we could do is we could switch this from this colavanol diphosphate compound with a single amino acid change back to the end-copulyl diphosphate, so the function in ghrelin biosynthesis. This is a precursor, which might give us actually already a good idea on how this enzyme and this novel function may have evolved from one of these original progenitors. But we can now also use just a double mutant here. We can introduce hydroxy functions, which is very interesting from an engineering point of view, or we can switch the double bonds around. And now we have sort of already a collection of several engineered enzymes that make it a lot easier for us to put these into microbial platforms to engineer them to produce these compounds. And this is actually what we did. What we're doing now is we're taking this whole collection of diterpene synthase and cytochrome P450s across about 15 or 20 plant species. We combine them in different ways. A lot of these enzymes are very promiscuous in terms of their substrate specificity, so we can combine diterpene synthases from different species with each other and can then co-express them either in microbial platforms or, again, we actually push a lot of work in Nicotiana benthamiana and can produce some of these scaffolds as potential lead compounds. These still, if you can't read it, these are still sort of at the low milligram range, so this is nowhere close to industrial scale. But what these platforms allow us now, they're ideal for us to now have good tools to go after the downstream pathways, especially cytochrome P450s, and move more efficiently through the whole pathway eventually. So this is sort of where we stand with our work on some of these medicinal plants. Another interest that we really started when we moved to UC Davis is a better understanding of the role of diterpenates in stress resistance in crop plants. And the one example that I want to show you here today is the diterpene metabolic network in maize. And this is actually, we launched this with support by the JGI, so this DNA synthesis grant, which was an enormous help for us to get started on this project. So it's actually a larger consortium. There's a number of people involved, and our goal really is to elucidate these terpene metabolic networks across a range of different species and better understand their diversity and the modular nature of these networks, eventually to go and develop a tool for what we like to call metabolic therapy of crop plants to enhance resistance in these species. So we're working on 6 different species right now. What we've done so far since we started last summer, we have identified about 30 terpene synthases and cytochrome P450s across these species. One major target that we had was a compound class here called correlexins, and there's another one. These are sesquiterpenates on the left here. They're called zealexins. The diterpenes are correlexins. These were found some years ago by Eric Schmelz's and Alisa Huffaker's lab, with whom we're collaborating on this project. And we were interested to find out how these compounds are made, because we do know that they actually are the major drivers of pathogen resistance, both in above and below ground tissues in maize. So we started to look into this. Again, first step was metabolite profiling. And interestingly, what we found was that these compounds are particularly abundant in the scutellum. So the part of the kernel that actually feeds nutrition to the embryo eventually, and we find these compounds quite abundant in these very early stages of maize development. So we isolated quite a bit of this scutellum tissue. Again, we did transcriptome sequencing and found 10 diterpene synthases here and just about 40 cytochrome P450s. And that's a good number. This is what we usually would expect in a grass to see this size of enzyme family. The next step, what we did, we looked actually in the scutellum across a number of days past germination to see interesting patterns of expression. And we combined this also with inducibility, here using one of the major maize pathogens, Cochleobolus. And we compared it directly to this enzyme here, AN2, which is a class 2 diterpene synthase that had been characterized already some years ago and shown to be involved in defense mechanisms in maize. And now we were interested in the next steps. And what we found here, we found 2 candidates that showed a very nice, similar expression pattern to AN2, both of which were inducible. So these were really prime candidates for us to look into. And what we found is a metabolic network somewhat more complex that one I showed you in Salvia divinorum. we actually have 4 different of these class 2 diterpene syntases. We have 5 class 1s. These actually work together in a sequential reaction to convert this GDPP precursor. And what we also found is that these 2 that we're interested in, KS2 and KS4, show quite a bit of substrate promiscuity, so they can interact with different partners of these class 2 enzymes, and then really are the entry point to diverse branches of diterpene compounds that are involved in crop defense. And so we're making our way through this network. Everything I show you here in yellow is what we've characterized so far. Here in red, these are still the steps we are working on, but we hope sort of by the end of the summer we'll have this network figured out and can really start working on how these different branches interact with each other. But just to highlight a little bit of this, so the characterization here shown is for our first candidate, the KS2, and we threw it in our co-expression system, so we co-express this enzyme together with the AN2 shown here in orange, but also with class 2 diterpene syntases that produce compounds that have a different stereochemistry, so plus CPP, as well as a syn CPP intermediate here. And you can easily see this the KS2 enzyme is very promiscuous and can produce a number of different scaffolds. Most important to us was this one here. So the combination of AN2 and KS2 produces end-isochorin, which indeed is the direct precursor to the correlexins. So this was a good example where we had a metabolite and we wanted to really go after the genes and could use sort of pathway prediction to find the first precursor. Now if we throw in KS4 in the exact same assay, it also is promiscuous. It can interact with all of these different class 2 diterpene synthase partners. But the products are, for the most part, different. With AN2, we actually only find one product here, which is this one here that we call dolabradine, structurally quite distinct from the end-isochorin. And it also can produce a number of different products using plus CPP and syn CPP. And so even with these few combinations, 3 different class 2 enzymes, 2 different class 1 enzymes, we can already create 10 different scaffolds that now can be functionalized by cytochrome P450s and further expand chemical diversity and defense capacities in maize. And so this was the next step for us. So we went back to our transcriptome analysis and looked for some of these P450 candidates. We found a lot of these that are involved in Limboa biosynthesis. We were targeting those that could convert terpenes. And we had 2 here, one that looked really, really nice, but it turned out actually to, as far as we know, not to be involved in terpene metabolism. The second best was this one here. And so we co-expressed this both in yeast and tobacco, and we could see when we combine it with AN2 and the KS4, we throw the P450 in, we can actually now take dolabradine, and it is a sequential reaction where it first attaches this very unusual epoxide group to the scaffold, and then here it's a C3, an additional hydroxy group. So now this is actually a completely new group of diterpenes that we hadn't seen before in maize. So we turned things around a little bit, where now we actually went with the enzymes and we found new compounds that we didn't know before. And when we looked back into the tissue, we actually did find dolabradine. And surprisingly so, we find dolabradine as well as the epoxide almost exclusively in maize roots. So it was a nice approach to really go from metabolite to gene, and at the same time, gene to metabolite to elucidate these networks. We can also show that using an AN2 knockout mutant, that we don't have any dolabradine left. This pathway is entirely dependent on AN2. And this was really interesting. Now we had a new pathway in roots, and we wanted to start and understand what it does. It being in roots, the first thing we did was to look into osmotic stress using sort of a mimic, using copper sulfate stress in roots. And we could show that both dolabradine, so the hydrocarbon, as well as the epoxide are highly inducible under osmotic stress. Correspondingly, the major diterpene synthase, the P450, are induced at the transcript level. So it shows that there seems to be actually some activity in abiotic stress defenses. But we also purified the compound, a few milligrams of it, and then used it for fungal growth assays in defined media. And we could show that the epoxide actually is very active in inhibiting the growth of 2 different Fusarium species. These are major maize pathogens. quite effectively in both cases. So it also shows pathogen defense properties. And this is something that I find very interesting, that we have this compound here that has sort of a dual functionality, both in abiotic and biotic stress defense in maize, and that's something we are really interested in digging in further now. So we generated a number of maize mutants in addition to AN2 that now will allow us to actually tap into different stages of these pathway nodes and network nodes to dissect which of these branches may actually be dominant in which type of resistance. And this is sort of where we're going. And really the JGI and the DNA synthesis we had here was crucial to be able to do this in a fairly short amount of time. So this leaves me to thank my group and all the students who were bold enough to join up with a new PI and go after these things. It's a fantastic team, and I very much enjoy working with them. A number of our collaborators that are involved in various projects, not all of them I could show today, and of course our funding resources that we could secure so far, first and foremost the JGI. So thank you very much, and I'm happy to take any questions. I think this one there.
Thank you for the talk. So I know that a couple of challenges that exist with heterologous protein expression systems like you're using are getting proper protein folding as well as substrate availability. So I'm wondering, did you encounter these challenges or other ones, and what did you do to overcome those barriers?
Yes, there definitely are lots of challenges. We can handle the terpene synthase pretty efficiently. So those are really usually not a problem. They're soluble enzymes. Once you start looking into cytochrome P450s, you're in trouble. They're membrane enzymes. They're usually located at the ER. And really, there are many groups who are working on overcoming this hurdle. I think what we can say is there isn't really one size fits all to express P450s. Some of our enzymes work beautifully in E. coli. Some others don't. That's part of the reason why we actually use yeast, tobacco, and E. coli simultaneously, to not actually fall into a time gap and be able to try and express these P450s as efficiently as we can. I wouldn't say we're there yet that we really can get to all of them. Some are really difficult. Another hurdle that we do encounter frequently is that if you, for example, use benzamiana, once you start hydroxylating your intermediates, we very often see them glycosylated. And that troubled us for some time until we just sort of randomly threw glucosidases in there, and there were our products. So benzamiana is well capable of dealing with foreign metabolites in that way, for example. So that's something to watch out for. But we're definitely still working on trying to find better ways to especially express the cytochrome P450s.
Hi. Do you know how stable your your last diterpene was that has the epoxy group on it? Or like once you put it into water, does it start to degrade because the ring opens or something like that?
The epoxide is actually surprisingly stable. So we did multiple NMR analysis on it in deuterated chloroform for weeks at a time, and it was completely stable. I have not yet dared to put it into water. But it's certainly an important point when we are starting to look at potential adjustment application to our plants to see how it actually could protect the plants. So it's something we're working on. Yeah. I think in water, stability would drop fairly efficiently.
Okay, so if there are no more questions, let's thank Philipp again.